With this, we would like to welcome Dr. Patrick Boyle from Department of Bioengineering, who's going to be talking today on integrating computational physiology and explainable artificial intelligence to improve health outcomes for cardiac arrhythmia. So Dr. Boyle is in the Department of Bioengineering and leads the Cardiac Systems Simulation Lab. His lab focuses on building models of human heart to cultivate knowledge about arrhythmia mechanisms and devise new treatment for these conditions. He did his BSc and PhD in his hometown, which is at the University of Calgary. He spent 7 years in Baltimore working with Dr. Natalia Trianova at Johns Hopkins, starting as NCSRC-funded postdoctoral fellow and eventually earning the rank of assistant professor there. In 2018, he moved to the Pacific Northwest with his family and is a fellow of the Heart Rhythm Society and serves as the associate editor for the Cardiovascular Engineering and Technology. He has been honored with multiple distinctions, including the Young Innovator in Cellular and Molecular Bioengineering Award and the University of Washington Department of Bioengineering Chairs Award. He has published over 80 papers in a wide variety of peer-reviewed journals covering topics ranging from basic science and computational methods in translational research. In his personal life, he enjoys learning new board games with his family. He is an avid hockey fan, and he has a large collection of excellent vinyl records, and he has solved the New York Times Daily Crossword for 1,176 consecutive days, and he's still counting. Welcome, Dr. Boyle, and we're really excited to hear about your talk.
So yeah, thank you so much for the kind introduction and for inviting me to participate in this exciting seminar series. I see lots of names in the participants list that I do recognize, and lots of names in the participants list that I don't recognize. And so this will be an exciting time to tell you about the research that we're doing in the CARDS lab. I will say it's a longer talk, so I put together an outline. I want to say up front, if you have any questions that just can't wait and you feel compelled to enter them in the chat, I'll try and do my best to catch them in progress. I don't mind being interrupted. By way of outline, I'm going to start by sort of going over the broad outlines of the clinical problems that we think about and focusing particular on atrial fibrillation, which is a cardiac arrhythmia that affects millions of people worldwide. I'm going to go through a computational physiology example that discusses, a lot of this was a sort of foundational work that I did during my time at Hopkins and that we've continued to pursue since then, focusing on whether we can use computational simulations to sort of custom tailor treatment planning for a particular type of procedure called catheter ablation, which is commonly used to treat many different types of arrhythmia, in particular atrial fibrillation. And then in keeping with the theme of the series, I'm going to start to expand upon, where does that leave us and how can we use explainable machine learning tools? The explainable aspect is something that I've really enjoyed sort of wrapping my arms around since I started my lab here at UW, and it's been really trainee driven. The work of a PhD student, Savannah Bafolco, who's now graduated and gone to work for Boston Scientific, it's really driven our passion for this emergent research area. And so I'll share two of her projects with you, one about predicting simulation outcomes from model features, so the anatomy of the model and the distribution of scar tissue in a cardiac model. And then the second example, which we're really excited about, which is unpublished work currently under peer review, looking at using the same type of machine learning, but to predict outcomes of clinical procedures. So for those of you who are less well acquainted with the cardiology aspects here, the heart is a multi-physics system. We tend to think of the heart as a mechanical pump, but there's a lot more to it than that. The schematic in this slide highlights some of the exquisite structures that underlie the bioelectricity of the heart, which has been the focal point of much of the research in my career. And as a sort of as part of the background here, I want you all to appreciate, those of you who aren't so well acquainted with cardiovascular physiology, that every single mechanical pumping event in the heart is necessarily preceded by a bioelectric excitation, which is encoded in the electrocardiogram, which most of you are probably familiar with, and that is sort of under the underlying biophysical events in the different parts of the heart are all associated with distinct electrical signatures. So this is a nice schematic cartoon from Medtronic that introduces the concept of bioelectric propagation. The heart doesn't all get electrically excited simultaneously. Rather, heartbeats initiate in a special region called the sinoatrial node, which is our body's natural pacemaker. Discrete heartbeats that originate from the pacemaker then travel through the right atrium and the left atrium, and that traveling is what we call propagation of an electrical wavefront in the context of bioelectricity. And once the atria are excited, propagation enters the lower chambers, or the ventricles, through this central area called the atrioventricular node and the His-Purkinje system that lines the inner surface of the ventricles. And that's what causes the heart to pump blood to the body. So critically, heartbeats can only travel from the upper chamber to the lower chambers at this one central point, the atrioventricular node that I mentioned before. And in healthy hearts, propagation can't happen from the atria to the ventricles across those red boundaries. And that's extremely important, because if the atria start to go haywire, they start to fibrillate, and that's a sort of quivering of the atria that is precipitated by chaotic bioelectric excitation. The AV node, the atrioventricular node that we discussed on the past slide, it serves as a gatekeeper that only lets through heartbeats occasionally. And this means that the pump function of the heart can largely be preserved. So blood still gets delivered to the body for the most part, but the beating becomes more irregular. So instead of being like a metronome, beating reliably around, 40, 50, 60, 70 beats per minute, the intervals between heartbeats become unpredictable and vary from cycle to cycle. So it stops being a metronome and it starts being more like, improvisational jazz. So this might make you think, okay, the heart's still beating, oxygenated blood is still being delivered to the body. This is probably not such a big deal. And that's somewhat true, but the problem is there's a tremendous association between atrial fibrillation and stroke. So in the quivering atria, blood can tend to pool up and linger for too long in the nooks and crannies, especially in the part of the heart, or the part of the left atrium that kind of hangs off the back and doesn't serve a important structural role, which is the appendage of the left atrium. And so when the blood pools up there, it can clot, and if the clot forms, leaves the heart, and becomes lodged in the brain, that's a stroke, which can be quite devastating. Now, this process itself sounds simple enough, but in fact, there are many things that we still don't understand about the complex relationship between heart disease, electrophysiological symptoms like atrial fibrillation, hemodynamic phenomena like blood clotting, thrombogenesis, and the departure of the clot from the heart to initiate a stroke. So the really scary part is that in many patients, the first sign of any abnormality, the first sign that there's any symptom at all, is the stroke itself, because people can experience atrial fibrillation without feeling any overt symptoms, without noticing that irregularity of the heartbeat. So in short, to sort of finish off the introduction, there are a lot of unanswered questions in arrhythmia care. The atrial fibrillation conundrum is really just one of them. But the entire premise of my scientific career and of the cardiac system simulations lab is that when we bring to bear computational tools, including machine learning and artificial intelligence, there's a lot of room for improvement. from those tools and technologies. So a little bit deeper on atrial fibrillation, the epidemiology and impact and our mechanistic understanding. It's the most, I hope. Audio still coming? Yes, okay, good. Please stop me or like do a hand raise or something if my sound or video ever disappears. Well, it looks like I turned off my video. I'm going to turn it back on, unless we want to keep it off.
No, you could keep it on.
Okay, I don't want you to miss the gesticulation that I'm doing, like when I was clapping earlier. Okay, so it's extremely common. The prevalence of AFib is on the rise. So these are pretty old numbers, actually, the 2.5-fold increase by 2050. I haven't checked back in the last 5 years or so to see where we're at on that estimate, but I would be shocked if we weren't meeting or exceeding that predicted trajectory of disease. A lot of patients transition from a form of atrial fibrillation that's paroxysmal, meaning the episodes come and go, and they last, anywhere from a half an hour to, a couple days. Many patients end up transitioning, if they've been in paroxysmal AFib long enough, to persistent atrial fibrillation, which is much more severe. And persistent atrial fibrillation, believe it or not, is characterized by episodes of fibrillation that last longer than a week without spontaneous termination. So this is a really long-standing arrhythmia. And in fact, there's even more severe forms of the disease where the arrhythmia is just considered long-standing, and people sort of live with that and try to keep the rate of the ventricular excitation under control. So what do we do in terms of treatment options? I alluded to this earlier, catheter ablation. It's a minimally invasive procedure. So essentially, the patient comes in, they end up getting catheterized through the groin, and the catheter goes up through the femoral artery. This is the inferior vena cava here, and the catheter is fed across from the right atrium into the left atrium. And then something is done, and I'm using intentionally vague language there, to destroy the tissue surrounding the openings of the pulmonary veins. And so in this example, it shows radio frequency ablation, but there are also technologies that involve cryoballoon to elicit freezing to create scar tissue, or more recently, and there's a whole lot of excitement about using pulsed field ablation to try and do this process really rapidly. But essentially, what the end goal of catheter ablation is to eliminate the source of the aberrant electrical activity that initiates episodes of atrial fibrillation. And so, kind of the gold standard is to go around the pulmonary veins here, where most of the triggers of atrial fibrillation tend to come from. And then depending on the clinician, the center, the trial, the patient, the manifestation of disease, there might be other areas ablated in the left atrium, or for example, in this case, they've done something called, I believe the clinicians will correct me if I'm wrong, but I think this would be called a cavo-tricuspid isthmus line to prevent further atrial flutters. It's not really important that you understand the intent or the purpose or the details, the nitty-gritty of catheter ablation, just the sort of general idea of the fact that it's done. And it's incredibly common. As of 2017, which was the best data that I could pull from the HRS website on short notice when I was making this slide, it was 75,000 ablations per year, but that number has undoubtedly gone up since then. And this matters because the long-term ablation success rates are still relatively low. And this is, it's sobering. For people with persistent AFib, the number hovers between anywhere from 40 to 60%. Long-term success rate, freedom from atrial arrhythmia after a procedure at 1 or 2 years. The relationship between the disease state of the heart itself and the reentrant drivers of atrial fibrillation. So this is something that I hadn't talked about previously, this sort of vortex-like activity that can be initiated during the course of atrial fibrillation. So this doesn't have to do with the trigger areas that I talked about previously, the pulmonary veins. Oops, sorry about that. But there's sort of a increasing understanding and recognition that the disease state of the atrial tissue itself could have a lot to do with the localization dynamics of these so-called reentrant drivers. Some people call them rotors, spiral waves, but these are self-perpetuating sources that can keep atrial fibrillation going once the activity from the pulmonary veins has gotten it started. But we really don't understand the nitty-gritty details of this. And once upon a time, when I was first starting to work in this area in 2014, 2015, our central premise was like, we could really get a lot out of mechanistic finite element models of the electrophysiology in terms of improving our understanding of this stuff. And so the starting point for the process that I'm going to talk about today is the acquisition of something called 3D late gadolinium enhanced MRI scans of the heart. And so in this case, the gadolinium binds with a higher affinity to scar tissue than to non-scar tissue. And so it shows up as hyper enhanced or brighter on the MRI scans. And so we can see those areas of structural remodeling, which are sort of the nexus of rotor perpetuation for those non-pulmonary vein atrial fibrillation driver sources. So we can contour the surfaces of the left and right atria, or sometimes, as we'll talk about later, just the left atrium. Then we can delineate those regions of scar, which are shown in green on the image in the bottom right. And then we can use different techniques like diffeomorphic mapping to deform the average human atrial fiber orientation. So these are the muscle fibers that underlie and have an important role to play in the dynamics of electrical excitation, but we can't acquire those through imaging yet. And so we kind of cheat and use atlas representations to get the nearest we can get to to something representing the underlying substrate for electrophysiological processes in an individual's heart. And that's the patient-specific model of the scanned atria. And we can then poke and prod and electrically stimulate that in any which way. So this is an example of when we rapidly pace the atrium. So this is a sort of imposed process. We don't actually simulate the true triggers that would happen in the wild from the pulmonary veins. We can imagine the initiation of atrial fibrillation episodes from any number of different areas within the atria through tachypacing. And it initiates these reentrant wavefront patterns. And so at this point, the reason why this is so important and a really fundamental thing to grasp here is that nothing is keeping the model going here. There are no external stimuli whatsoever affecting the evolution, the spatiotemporal evolution of the electrophysiological propagation here. We're just seeing wavefronts of excitation that are self-perpetuating and that'll keep going until the cows come home, because of the presence of a reentrant driver, in this case up here in the vicinity of the superior vena cava. And so one way to visualize that is through these four-dimensional movies on the left, and another way, which I'll use throughout the presentation, which is a little bit more succinct and concise, and we can publish it in papers because we can't do videos, of course, in those, is to collapse those into an activation map where you see this sort of typical pinwheel pattern surrounding what we would call the sort of organizing centers or phase singularities of that reentrant driver. And so that kind of slug shape, that's what we would consider, like if we want to prevent that particular reentrant driver in the context of this model from perpetuating activity, one thing that we could do is, envision or simulate the ablation of that region, because if it's no longer there, if it becomes scar tissue that can't conduct excitation at all, then obviously the arrhythmia can't be perpetuated in the model. And so, back in 2016, the really key finding that emerged from our original mechanistic work in this area was that each individual patient, so we took a population of 20 MRI scans from different persistent atrial fibrillation papers at a center in Bordeaux, France, and each of them had sort of a specific subset. No matter how much we paced and prodded and poked the models in all sorts of different ways, the reentrant driver activity always ended up being in 1 or 2 or 3 or maybe 4 specific regions, which is an exciting finding in this context because it made us realize, like, well, maybe if we came up with a strategy to, isolate the pulmonary veins to get rid of the triggers, and then get rid of these areas to destroy the potential substrate areas for perpetuation of the arrhythmia in persistent atrial fibrillation, then we could have like a truly effective custom tailored treatment plan. And so a couple of years later, that's exactly what we did. And again, this is a huge collaborative project, and I wrote the paper, and I'm the first author on it, but it was the work of well over a dozen really talented clinicians and bioengineers and imaging specialists at Hopkins, and so I want to pay appropriate tribute to them here. And so what did that look like? So like I said, we get these pre-procedure MRI scans to characterize the shape and distribution of fibrotic tissue. Then we reconstruct models and we run simulations to identify those areas that I've spent the last couple slides talking about, those regions that are most likely to harbor reentrant activity. And then we use clinically radiofrequency ablation to strategically eliminate those rotor trajectory areas from each patient's fibrotic substrate. So that's the catheter ablation procedure I talked about previously. An interesting wrinkle is that one of the things that we decided to do was, after we computationally or virtually did the ablations, we wanted to make sure that we were well and truly done. There's always a possibility that after an initial ablation, new targets that were previously latent or masked by the presence of more obvious places for rotors to localize would emerge. And so we did sort of a rinse, lather, repeat process in the inner part of this workflow before we ultimately concluded that a model was completely non-inducible, and then exported that ablation plan, that patient-tailored plan, to the CARTO system, which is the electroanatomic mapping system used during procedures, so that clinicians can keep track of where their catheter is when they're about to deliver, RF or cryo or pulsed field ablation or whatnot. So here's an example for a case, a patient who came in 2017 in June to Hopkins, presented with persistent AFib, needed an ablation. The problem here was that when we ran simulations, we never saw any of those rotor locations at all. The particular geometry and the distribution of scar tissue in this individual was such that the only thing that was possible to induce was what we would call an atrial flutter. And so this is reentry. You can see it's a little bit hard to make out on these maps as I'm screen sharing, I'm sure. And I'm trying to use the cursor to show you, but we could see either clockwise reentry around the mitral annulus. And so this would be the area of tissue immediately adjacent to the left ventricle, or counterclockwise. But we never saw any of those juicy rotor targets that we were looking for. And so our solution was, well, let's digitally ablate it. Let's virtually ablate that. And so we put a big, thick line of dead tissue, which is represented in the gold regions here in the activation maps. And lo and behold, we observed the initiation of a bona fide reentrant driver here in the inferior part of the posterior left atrium. And there's another example of a similarly located rotor a little bit farther to the right, closer to the right inferior pulmonary vein over here. And so that's the kind of thing that gave us this idea, okay, well, we can export those, we can draw those lines onto a map, and we can export that to the mapping system that's used during the clinical procedures. And then on the day of the procedure, the patient's on the table, and the clinician, who in this case was Hugh Calkins, one of the attendings at Hopkins, went in with the catheter and tried his best to follow the paint by numbers that we gave him. So at the beginning of the procedure, the patient showed up actually in sinus rhythm that day, but they were able to pace the patient into atrial fibrillation that was durable. It lasted about 30 minutes before we decided to start doing any ablation. And during the ablation of that target, that sort of check mark shaped thing that you see on the electroanatomic map here on the right, right as soon as Dr. Calkins got to the end of that check mark shape, lo and behold, the arrhythmia terminated. And so you can see it goes from chaotic activity over here on the left to termination on the right. And no matter what after that, for the duration of the procedure, it was impossible to reinduce atrial fibrillation through pacing from the coronary sinus. And so this is a pretty compelling thing to see happen in the wild. And I have to emphasize, this isn't the be all end all. Like acute termination during a procedure, for those of you who are electrophysiologists or cardiologists, you're probably nodding your heads right now saying like, well, okay, it's nice to see in the lab, but it certainly isn't the answer. We know from long-term studies that this isn't necessarily indicative of a lasting freedom from atrial fibrillation. So this could be smoke and mirrors. And so we needed to follow up and look at the effects in the entire pilot cohort. So we were able to do the analysis in 9 out of the 10 patients. And again, I tend towards a really conservative appraisal of these results. Some of these patients we followed up for quite a while, over a year. Some of them were a little bit over half a year. But generally speaking, we were cautiously optimistic about the outcomes. There were no recurrences of persistent AFib. Only one of the patients was back in paroxysmal atrial fibrillation with a little bit of atrial flutter at the end of their follow-up. And this is now being tested prospectively at Hopkins in a trial that I'm still sort of tangentially involved with. So key takeaways from the part of the presentation about computational physiology. I hope that I've convinced you that there's sort of proof of concept here for a new treatment paradigm. We are cautiously optimistic about the clinical outcomes, but they really need to be validated as part of a prospective and randomized controlled trial, which is now ongoing at Hopkins. But the thing that will launch us into the second part of the talk is that what this means is that as we become capable of doing these types of large computational physiology projects, we're going to have larger data sets, and the larger data sets create an opportunity for us to use artificial intelligence and other machine learning approaches to learn even more about, why do these arrhythmias happen in the first place? What can we do to better treat them, and how can we navigate the process of helping people understand their own disease? So the premise of this part of my talk is going to be, and I don't think I need to convince anybody here of the fact that AI and machine learning are powerful but imperfect tools. And there's a laundry list on this slide of examples of people kind of tripping over their early enthusiasm for AI and machine learning. There's the requisite story over here about how ChatGPT, I can't remember if it was slander or libel, but it did something. It falsely accused a mayor of some Australian town of being a criminal when apparently he was actually some kind of brave, heroic crime fighter. But there are all sorts of limitations and guardrails that we need to think about in the pursuit of artificial intelligence and machine learning, and especially in the healthcare world, we really need to be cognizant of like, okay, is this hype or is there actually hope at the end of the day for adoption of these tools in a responsible and effective way? And so, like I said earlier, in the late stages of the lockdown phase of the pandemic, I had a PhD student, Savannah Bofoco, who pushed me really hard because she really wanted to do more machine learning research, and I was skeptical for many of these reasons. But she said, "Listen, Pat, like if we can do our part to improve trust in artificial intelligence and machine learning in the context of cardiovascular research, especially electrophysiology, then there's real promise here for success." And so she convinced me to buy into the hype a little bit by pointing out to me the fact that we're a lab that's really well equipped to try and increase transparency of the computational tools here. And so she taught me about all sorts of emerging tools for explainable machine learning. There are a number of different things that are sort of called out on this slide, which Savannah helped me make. But the one that we settled on was sort of a homegrown, and I think that probably all of you at this seminar series will be familiar with it at this point, is the Shapley additive polynomials technology. This is homegrown from Sue and Lee's group in the computer science department here at UW. And the way that Savannah pitched it to me was, okay, you got to think about a model feature and its features like players in a game. And what the Shapley polynomials help us do is characterize the extent to which each player, so each feature that we feed into the model, is going to contribute to the outcome of the game. So are we going to win or are we going to lose? And so I said, all right, that seems like an interesting enough analogy. Can we put this into a slide that I can present when I'm invited to talk about it? And she said, sure. You're always complaining about hockey and telling us about how terrible the Calgary Flames are. So this goes back to my roots in Western Canada. And this is still true, by the way, they're still terrible. And so let's talk about what's going to add to the incremental value of what's the incremental value that each player adds to the lineup if the outcome that we're looking to predict is, are we going to lose the game or are we going to win the game? Okay, so will our team win? So I took, I scraped some data from the NHL statistics page, and I came up with, okay, this is one of my faves. This is Nazem Kadri, power forward. Okay, this guy was new at the time when I made this slide, Mackenzie Weegar. I still think he's a bit of a stud. He can do better than that. He's contributing. He's pushing the team in the right direction. Now, this guy, Chris Tanev, some of you might be familiar with his brother, Brandon, who plays for the Kraken. Bit of a boat anchor on our lineup. Hopefully we'll be trading him soon. And then you've got old Milan Lucic, and he was pulling us down. Fortunately, since I made these slides, he's been traded. So there are 4 players here. We need a 5th player. And so for illustrative purposes, let's see what would happen if we added me on right wing. Okay, there's me rocking number 82. I am terrible at hockey. I can barely skate, let alone skate backwards on an NHL rink. We are going to lose badly because my contribution to the game pulls us far, far in the wrong direction towards we will lose. Now, what about my little brother, Brendan? Okay, he's better than I am. Maybe we win. Okay, and so the idea of Shapley analysis, as I sort of crudely illustrate here, is you take all the factors and you can assign on a case-by-case basis how much each factor, if you add up everything that's pulling the model in one direction, and you compare it to everything that's pulling the model in the other direction, you can get an overall estimate of, how likely is the outcome that we're interested in. So in the context of modeling and simulation of arrhythmia, we can look at other interesting problems with technology like Shapley. So we have these enormous imaging data sets that have yielded model populations that are really ripe for mechanistic analysis and population scale analysis. And we can systematically look at these simulation data sets in ways that are challenging with conventional tools. So let's look at a couple examples, and then that'll be the end of my presentation, and we can move on to Q&A. I think I've been going for about 35 minutes, so I think I'm on track here. So one thing that we might be interested in is, can we predict the recurrence of atrial fibrillation following a catheter ablation procedure? So there's a difference between this map and the map that I've been showing you on previous slides, which is the presence of these ablation lesions that are actually image derived. So in the Optima study that I showed you earlier, all of the ablation lesions that we simulated were essentially fabricated. Those were virtual simulations. Those were the sort of, the fanciful visions of the computational modelers. In this particular data set that Savannah and I worked on together over a period of several years with our collaborators here in cardiology, we had access to 2 sets of MRI scans per patient. So the first set of MRI tells us where these green regions are. So it tells us the geometry, and it tells us the pre-ablation distribution of diseased tissue. The second set of MRI scans tells us something completely different. It tells us the locations that have been rendered well and truly non-conductive by the ablation procedure itself. So the ablation-induced scar tissue. which is whereas the green tissue is it has compromised electrophysiology and structure, but it can still conduct impulses. The gold tissue in these examples is considered to be well and truly dead because it's been touched and essentially cauterized, burned away, rendered non-conductive by the ablation process. But we know that a lot of people who receive an ablation like this still recur, about 30, 50, 40, sometimes as much as 60%, depending on your study of choice. And one hypothesis is that pro-arrhythmic substrate created by the lesions themselves might play a role. And so Nazem Akoum, our collaborator here at the University of Washington, has a paper from a few years ago that looks at the interaction of this sort of residual scar, residual fibrosis, with the ablation-induced scar. And the idea here is that if you create scar in the wrong place, it could unlock new substrate, or it could create an area where a wavefront could sort of ring around the rosy around ablated tissue and cause a new problem that could lead to arrhythmia recurrence. But this is poorly understood. We've characterized, we have a moderate degree, I'd say, in the field of understanding how pre-ablation fibrosis affects the propensity for arrhythmias in atrial fibrillation. But we wanted to better understand how post-arrhythmic substrate works. And so we looked at a large data set here. So we had, like I said, both pre-ablation and post-ablation models in the same patients. And what we found was that indeed the models recapitulate the behavior that we know or that we hypothesize exists in patients, which is we can see the reentrant patterns propagating around regions of scar. But this wasn't universal. So, for example, in this case, or in these two cases that are illustrated on this slide, there were a lot of different areas of scar. You can see them as the different gold regions up here in the maps that show just the anatomical and structural data. But not all of those scars are created equal. Only a subset of those scars can actually sustain post-ablation arrhythmia. So, for example, the two scar regions shown in these maps on the bottom, I've shown you the examples of how they can sustain reentry in the computational simulations, whereas the other ones cannot. And so what we wondered is, with this large data set comprising 37 patients with 2 models each for a total of 74, we identified 633 distinct scars, and 90 of those were pro-arrhythmic. 90 of those were the magenta-coded ones here that could actually sustain a post-ablation scar-dependent arrhythmia. So in this context, when we reframe it for Shapley analysis, the game is, will a particular ablation scar be pro-arrhythmic in the model? And the players are the features of the lesion. So the proximity to residual fibrosis and the size of the region, or of the lesions, is what we focused on, these ablation areas. And so reframing the SHAP analysis from the hockey problem, we're now thinking about how do the different spatial features of a particular scar lend to its being classified as either pro-arrhythmic or not pro-arrhythmic. And so Savannah did some very tedious and difficult work to try and tease out all of the details of the different spatial features that we could use. But at the end of the day, the top line result is pretty good. We can do a fairly decent job. And this is using random decision forest. So it's the most, I don't want to say it's the most simple possible form of machine learning, but it's a fairly rudimentary form. But the augmentation with the Shapley analysis is what really allows us to go farther here. And so I don't have the other statistics to describe the predictive accuracy of the model on hand, but I can assure you that the accuracy and F1 score, precision recall area, were all reasonable and consistent with the nice ROC curve that showed around a 0.9 area under the curve, suggesting that we could predict which islands of scar were more or less likely to perpetuate these post-ablation arrhythmias. And what the Shapley analysis allowed us to do was identify the particular regions of interest. And so in terms of the size of the scar, and this is where Savannah and I put our heads together again to do some nice scientific narration, what we saw was the presence of a sort of Goldilocks phenomenon. So scars that are too small, so that you have either too little perimeter or too little area, are unlikely to perpetuate arrhythmia. That's fairly reasonable. And scars that are too big are also a little bit less likely to perpetuate arrhythmia. This is probably because the wavefront, by the time it gets all the way around, it's not going to have time to re-excite, and other factors are going to interfere with its ability to serve as an anchoring point for those arrhythmias. In contrast, there are lots of non-conductive obstacles. So we considered the mitral annulus and the pulmonary vein sleeves just the same as a scar in this context because they're functionally indistinguishable. So this would be the size and perimeter that would be just right in that sort of Goldilocks area. Interestingly, and consistent with our hypothesis, proximity to this residual fibrosis, so that's the regions that were identified on the original pre-ablation MRI scan, that's also bad. And so in this case, we have a case on top, or in this slide, we have a case on top where there's very little residual fibrosis versus on the bottom where there's a lot of residual fibrosis surrounding the highlighted lesion. And indeed, in this case, the surrounding the lack of surrounding residual fibrosis here pulled the model towards a decision, a sort of equipoise between whether it thought that the scar would be arrhythmogenic or not. And in fact, it was, but this turned out to be kind of a borderline case. Whereas in this case, the size criteria were both pulling the model towards saying, no, I don't think it's likely for there to be reentry around that scar. But it was eventually steered towards the correct conclusion by examining the surrounding residual fibrosis and one of the other factors that we looked at, which was the proximity between distinct islands of ablation scar. Okay, so the key detail here is that the Shapley can give us deep insights on cases where the classification is borderline because of contradictory factors. In both of these cases, the geometry says one thing and the presence of residual fibrosis says another. In both cases, the model got the prediction right, but it was only because of the inclusion of the different factors that we were able to arrive at that conclusion. I don't know why there's a big yellow cloud on the bottom of this slide. I think that's an error on my part. But the plots here basically show the quantitative data, looking at the area, perimeter, and the presence of residual fibrosis as a function of the SHAP values as a function of those parameters. But the important caveat here is that I need to highlight, and I tried to telegraph this in the outline. This is using ML to predict simulation features from computational model features. And so this isn't a true translational application yet. And so to conclude my presentation, and I think I'm still relatively on time here, I'm going to talk about our attempt to do just that. What if we took all of the information available to us for these patients, the pre-ablation MRI, the post-ablation MRI, the clinical attributes, the simulation features, and also information from their electronic health records, so their medical history, their demographics. What if we took all of those together and tried to look at the possibility or at the likelihood that the patient would have a recurrent arrhythmia following an ablation procedure? Okay, so this is a pie chart that's made up. But what it's meant to communicate is the idea that there are many, many different factors, even from a clinical context, that are considered possible drivers of recurrence after a failed AFib ablation. So it could be because of comorbidities. It could be because of structural changes of the heart from the ablation or from other processes. It could be because of post-arrhythmic substrate created by the ablation, like we just talked about in the previous study. It could be because of pulmonary vein reconnection. So sometimes the tissue that's thought to be rendered ablated by the ablation catheter ends up apparently healing, and the arrhythmia can be initiated again by pulmonary vein ectopy. And so what we're curious to know is, can we put a quantitative tag on any of those things? Figure out how much each mechanism is likely to impact the actual likelihood of recurrence, and maybe identify some population level trends to broadly classify individuals who've had an ablation, where we have the MRI scans from before and after the procedure, to say, we think this person is likely safe, likely at risk, or somewhere in between. Now, the list of features available is enormous, and this is just a subset of them. One of the things that working sort of shoulder to shoulder with the clinicians really gives us a privilege for is that we can take this list directly to our clinical collaborators and say, hey, like, is anything up here? And in this case, we took it directly to Nazem and his team, and Nazem said, well, we might not want to use all of those features because some of them don't really make sense. Some of them are unlikely to be dispositive in the context of AFib recurrence after an ablation procedure. And so what we settled on was looking at fibrosis features from simulations. We looked at the region-wise distribution of fibrosis in the left atrium before and after the arrhythmia, the ablation. We looked at the locations and features of simulated AFib drivers from patient-specific simulations. So that's the computational physiology angle, although, spoiler alert, we ended up throwing those out, and I'll talk about that a little bit at the end. And pro-arrhythmic ablation scar from the Shapley analysis that we had previously done on the, predicting model simulation features from model attributes. So the predictive power of this clinical application of the RDF with Shapley was not bad. So it's a little bit more modest in terms of the area under the curve, and the other metrics follow, the F1 score, the PR curve. All of them are more or less consistent, and we're fortunate to have like a relatively balanced data set, which means that we can usually get away with showing just the the ROCs. And so that number, 0.84, is consistent with what other groups have reported for similar studies without the benefit of the explainability analysis, with slightly different imaging techniques, but with completely different patients. And so this is a paper from the Hopkins group from 2020 that reported an AUC of 0.82. This is a paper from King's College of London, group led by Caroline Roney and Steve Nieder, that reported 0.85 for a similar classification problem. And so we were happy that we were in the right ballpark, but then we got to go to the really exciting part, which is like, let's dig into the details and look at this rank ordered feature list. So what is it about the features that we know that can help the model determine whether a particular individual is more or less likely to be at risk for a recurrence following their ablation? And so the bee swarm plot here tells an interesting story. I don't want to go into all the details here because we would be here all day, but essentially what this draws our attention to is the fact that the left atrial volume index after the ablation, the number of distinct areas of ablated tissue that were created that we can see on the post-ablation MRI scan, the post-ablation residual fibrosis in certain regions. All of these are sort of our leading candidates for classifying individuals in one direction or the other. So the approximate way to interpret this would be the sort of usefulness of the feature overall across all of the cases that were examined in the Shapley framework. The key relationships that sort of tumbled out of this process were that having a higher, like I said, having a larger atrium following the ablation procedure tended to be predictive of arrhythmia recurrence, and having more distinct areas of hyper-enhanced tissue that represent the areas that were ablated and durable enough to show up on the post-ablation MRI scan 3 months after the procedure. Having more of those made you less likely to be classified as a recurrence, or as at risk for recurrence. But to me, where the Shapley really shines is in these arrow plots that sort of give a patient-specific breakdown of how every single feature that was ingested by the model factored into a particular decision. And so to me, like as a engineering research in this area, and as frankly as a family member of a person, my dad who suffers from atrial fibrillation, this is the kind of thing that I want to see. And I think that it's important to try and strive for this level of accountability if we're going to deploy machine learning and artificial intelligence. And we're going to say, okay, well, we gave you a 60% likelihood of recurrence. I think it's natural for people to ask the question, well, why? Like, what does that have to do with? And so in this case, you could tell the patient, you could say, okay, well, the model thinks you're likely to recur because of the residual fibrosis in your left pulmonary vein region and because of the amount of scar that we put in the right pulmonary vein region, which was relatively low compared to other patients. And these arrows have different lengths and different, and in some cases, different directions on a case-by-case basis from patient to patient. And so it's really revelatory for me to be able to look into each individual decision that the model has made and really sort of show your work and say, here's how we got there. There's inter-individual variability in the role played by each feature. That's kind of saying in pictures what I just said in words. And so this is where we're at with this project. As I said, this work is under review right now, and so we're hoping it'll be published soon. But we're really excited about the potential impact that this could make and about how we could take it further from here. There are lots of different ways we envision using these. Because I'm running a little bit long on time, I'm not going to go through each of these point by point, but the one that I tend to focus on is really what I just mentioned. And I tried to give the sort of anecdotal example of, like, you know, sometimes we have this conversation like, well, do patients really want to know what's inside the black box? Or do they really just want to get better? And like personally, and this is a values thing, personally, I think that, you know, there's going to be variability, but I think that it would be good for us to put ourselves in a position where we can share that information if the patients and their loved ones are interested in hearing it. And so that's where I see the power of explainable machine learning, especially in contexts like these. So wrapping up, we think that XML can be really useful in helping to untangle mechanisms in computational models. And the example I showed you was for proarrhythmic features of ablation lesions. Prediction of clinical outcomes in AFib can really be clarified and crystallized with explainable machine learning, and we think it's a useful tool in the context of deploying machine learning in clinical settings. And it's important to recognize, as part of this process, this is sort of a corollary, is that apparent individual drivers of recurrence can vary quite dramatically from patient to patient. And so just taking out the population scale feature is not good enough. You can't necessarily make good decisions based on like your top 2 or 3 criteria. So to revisit just finally the sort of premise that I introduced at the beginning of this section on machine learning, like, is there hype or is there hope? And I think the answer that we've arrived at is that there's a little bit of both. And we're hopeful that with judicious use of emergent machine learning tools, we can help have more of the latter than the former. So this is my lab, and these are the people who did the work. And thank you so much to the organizers of this seminar series and to the funders who contributed so much to the studies that I've shown today.